Hello everyone, welcome to the webinar. I'm Lizzie Segoo, I'm President-elect of the British Society of Soil Science, and I'm delighted to welcome you to our 6th webinar of the year. Before I welcome our presenters, I'd like to introduce the British Society of Soil Science as hosts of today's webinar. We're an established international membership organisation and charity committed to the study of soil in its widest aspects. We bring together those working within academia and have a growing membership amongst practitioners implementing soil science in industry and those with a keen interest in soils. So we plan to host 10 webinars during 2025, so keep an eye out on our website for further details. Before we kick off, just some basic housekeeping. As there's so many of you here today, all your microphones have been muted. We will be taking questions at the end of the presentations and we'll be monitoring these throughout the webinar. So please could you try and submit any questions that you have via the questions box by 12:50 to give us time to get through as many as we can. Today's presentation has also been awarded BASIS and Neurosis CPD points. So if you're registered with either of these schemes, please contact us directly after the event and we'll allocate you those points. Finally, please be aware that we're recording today's presentation. So the webinar today is organised in partnership with the British Society of Soil Science corporate member, Rothamsted Research. Rothamsted is the longest running agricultural research institute in the world with a proud history of groundbreaking discoveries. Rothamsted is home to 3 unique resources open to researchers from all over the world: the long-term experiments, Rothamsted Insect Survey and North Wyke Farm Platform. So to tell us a bit more, I'd like to introduce Impact and Engagement Manager, Dr. Georgia Mitrosa, to briefly explain a bit about Rothamsted.
Amazing. Thanks very much for this introduction, Lizzie. Good afternoon, everyone. Just a short introduction about Rothamsted Research before we start with the presenters of today's webinar. So can I have the first slide, please? So Rothamsted Research is a globally recognized institute at the forefront of agricultural science and innovation, and is dedicated to addressing critical challenges for the food production and environmental protection. It is one of the oldest agricultural research institutes in the world, having 2 campuses and 2 additional field trial sites with 830 hectares of field environment. It is a world-leading non-profit research center that focuses on strategic agricultural science to the benefit of farmers and the society worldwide. Currently, there's more than 500 people employed from different nationalities, and we also have collaborations with different countries abroad. We're very actively producing scientific publications with about 300 publications per year, and simultaneously we have about 300 ongoing projects at the same time. Overall, we have active engagement with the farming industry and the public to deliver knowledge, expertise, and research outcomes. So on the next slide, please. Rothamsted Research Foundation dates back from 1843, when John Bennett Laws, the owner of the Rothamsted estate, appointed Joseph Henry Gilbert, a chemist, as his scientific collaborator in order to together first find out what was around this scientific collaboration, and planted the first of what are become to known the classical Rothamsted long-term experiments on broadblock field. So they help us to form and develop our understanding of plant nutrition and produce the world's first commercial chemical fertilizers. On the next slide, please. Nowadays, we have moved on from that, and at Rothamsted Research, we work on a lot more than just that, including high-tech monitoring of insects, the importance of field margins for the abundance of pollinators and in general beneficial insects. And we also look at the life underground in the soil, and we do this with a combination of field and lab work and by using molecular tools. So this brings together fundamental and applied research with real world impact. And the next and final slide, please. As already been mentioned by Lizzie, Rothamsted is home to 3 unique capabilities open to researchers from all around the world. So this is the long-term field experiments set up 182 years ago, which is the oldest continuous agronomic experiments in the world. But nowadays, we also are asking current questions and applying new methods that we gain novel insights on more than agricultural challenges from that. We also host the National Insect Survey by running 2 trap networks in the UK since 1964 to monitor aphids, large moths, and many other migrating insects. Also not to forget the Northwick Farm Platform in Devon, which is a grazing research facility and that focuses on the environmental impact and efficiency of the livestock production nowadays. It's a very short introduction to Rothamsted Research. Obviously, we're doing a lot more than what I was able to present today, but I leave this there for now. And if you have any questions or specific requests, please do reach out to us. Thank you.
That's great. Thanks, Georgia. So now I'd like to introduce our presenters and invite them to join me on screen. Our first presenter today is Professor Jon Storkey. Jon is an agroecologist with a background in sustainable weed management. He's been following with interest the growth in regenerative farming practices and is interested in how the concept can be defined and the benefits quantified. He's leading on a new long-term trial at Rothamsted that is monitoring the effects of reduced tillage, organic amendments and crop diversification on soils, crops and biodiversity. So Jon will be presenting today with Jonah Prout. Jonah's a postdoctoral researcher at Rothamsted Research with a focus on soil carbon. He's currently working with and further developing the Rothamsted soil carbon turnover model. Jonah's PhD investigated soil organic carbon clay ratio as a metric for assessing soil carbon status of agricultural soils linked to soil structure. And then our final presenter is Dr. Jenny Bussell. Jenny is a soil scientist with a background in greenhouse gas emissions, soil health and carbon sequestration. Jenny currently runs the agroenvironmental research at the Allerton Project, which includes agroforestry, biostimulants, biochar, and a long-running trial on both the profitability and soil health aspects of regenerative farming. So thank you all for agreeing to present today. So we wanted to start our presentations by posing a question to you all, and that is, what do you think regenerative agriculture means? So to our audience, please feel free to add your definitions via the questions box as well. But Jon, we'll come to you first.
Thanks very much. Thanks for the opportunity to speak this afternoon. So again, these are personal perspectives, but I think regenerative farming is a continuum, not a category. So it is a very useful framework for interpreting different combinations of practices in different circumstances or contexts. So there's nothing particularly novel about any one of those practices, whether it be reduced tillage or cover crops or crop diversification. And they might previously have been given other labels like agroecological approaches. But it's a useful framework for looking at novel combinations and how we can move systems along that continuum. So my ambition and goal wouldn't be that we have a subset of farms that we can call regenerative, but that we're moving all farms along that continuum.
Yeah. Jonah, did you want to add anything to that, or is it you, Jenny?
Yeah, I don't mind, sorry. Yeah, I think, well, I agree with Jon. Yeah, that it's sort of that continuum and that you pick different aspects of it depending on context. And it sort of, well, I have a slight bias, but it feels sort of like a soil first approach to farming effectively. So understanding that that's sort of the foundation of the industry and, you know, our lives and food production, and really working towards building that up as the resilience of that sector effectively.
Yep. Jenny?
Yeah, I agree. I agree with my other presenters, obviously. And I very much agree with this, the soil first element. I think it's not a recipe of the things you have to have on your farm, but it's an aim. It's an aim to make your soil in a better state than it was. And that will fluctuate depending on what you're doing, what crop you've got, what the weather's done to you in the last few months. But it's that overall goal and the decisions you're making with the soil health and that improvement in your goal and in your mind.
Yeah. Okay. thank you all. I think that set the scene nicely for us to lead into our first presentation. So Jon, we'll pass over to you now.
Okay, so I'm going to assume that's working.
Yep.
If I don't hear otherwise, excellent. Okay then, so it's a very short talk that I've got this afternoon. I'm going to present a new experimental platform that some of you may be familiar with already, but has been designed to address some of the complexity around this concept of regenerative agriculture that we're spending the next hour or so thinking about. And as I started to allude to in my introductory remarks, there's two big challenges, I think, to the concept. One is the one of categorization. So at the moment, the community is wrestling with this question around certification. Can we certify a farm or a system as being regenerative? And it's very difficult to do that because Jenny's right, it's not a prescription. There's not a one size fits all that you can cut and paste into different contexts, but it's the way I think of it is alternative recipes or similar ingredients in different combinations that give you different flavored cakes or size cakes or whatever. So I've just picked out some of the example ingredients here that you can have in arable systems and in livestock systems. And obviously one of the principles of Regen Ag is to move towards more mixed systems that integrate livestock into arable. But you can't say that that farm now is regenerative and that one isn't. So that's the first problem that we're wrestling with at Rothamsted in trying to provide the evidence base. The problem of categorization. How do you quantify the metrics of a system along this continuum of regenerative transformation? And the second problem, and Jonah will talk about this in more detail, is contextualization. What works where? So the reason a single recipe or a single prescription cannot be rolled out to transform the whole of the UK to regenerative agriculture is some things work well somewhere and other things work well elsewhere. And the classic example of this is soil texture and the response of soil organic carbon to soil texture. So these are a couple of examples from our existing long-term experiments that compare the buildup of soil carbon over decades under contrasting land management. Woburn is a sandy soil, and so we see very little response to the treatments, whereas Highfield is our clay field, clay soils at Harpenden, and so we see a very strong response to the land use change and specifically the arable reversion. So we have these two, I think they're fundamental challenges to the research community seeking to feed in evidence to the Regen Ag conversation. Categorization, how do we compare a regen system? How do we define it? And contextualization. And that makes for a very complex research question, very complex research field. It's not as simple as just demonstrating that a particular new fertilizer affects yield in a particular way. It's more of a Rubik's Cube. It's more of understanding how almost an infinite combination of different contexts and combinations of practices will impact multiple outcomes. And I took this giant Rubik's Cube to Groundswell last year and just asked people to write on it what their priorities were for research into Regen Ag. And this just gives you a snapshot of that, an overview of that, of what people's priorities were. But I've just put this slide up to illustrate this concept of complexity, that we're talking about very complex systems. Jenny will talk about the impact of seasonal weather as well, and these kind of jokers that are played into the pack as well. So how do we design experiments and how do we design research programs that can help us navigate this complexity? And Rothamsted is ideally placed to provide that evidence base because what you need are long-term experimental platforms that can be used to unpick this complexity and provide some predictive power to say that if you did a certain thing in a certain farm, these are the responses you would expect to see in different outcomes. And that was very much the vision for the new regenerative farming experiment that we set up at Rothamsted at 2 sites. So we're fortunate in that we've got a research farm in Suffolk at Brooms Barn with a light sandy soil, and our main campus at Harpenden with a heavy clay soil. So we can look at those different contexts. And then we set up this experiment specifically to understand how different outcomes co-vary. If we pull a particular lever like reduced tillage, how does soil carbon respond, but how does weeds respond? How does soil microbiology respond? So the experiment was designed to look at multiple ingredients of Regen Ag in systematically different combinations, and then to monitor the trajectory of these different outcomes in the circles there. So biodiversity, soil health, pest weed disease pressure, greenhouse gas emissions, yield and quality, for example. So this slide introduces the structure of the experiment. We have at both sites a total of 60 main plots, and each of those 60 plots is in one of 3 rotations. We have a very simple rotation with 2 winter wheats and North Sea rape, a more complex one with 2 winter wheats, a spring barley and a spring beans, and then we have the more complex rotation with a perennial legume-based lay in it as well. So that's the principle of crop diversification, of green cover. Then we also introduce a tillage treatment. The plots are either in a ploughed system or in a reduced till system, ideally zero till. So that's your reduced disturbance principle. And then each of those 60 plots is split into 2, and on half of them we grow cover crops and we add organic amendments, green compost, and on the other half we don't. And then finally, so that's the improving soil carbon principle. And so, and then finally, we have a treatment that is reducing pesticides, reducing inputs on half of the plots and not on the others. And the beauty of the experiment is that it combines all of these ingredients in different combinations. So we can look at the effect, for example, of cover crops in a zero till system or in a plough system. We can look at the effect of compost in a diverse rotation or a simple rotation. And I think that's the real research need, and the evidence gap is how these different practices in the context of regen principles interact and impact different outcomes. So I won't dwell on the detail of the rotations, but they've been designed to be flexible because the experiment is long term, and so we have the flexibility to slot different crops in and out and change these up to a degree. But this illustrates the fact that we have a diversity of cropping, and then we're also introducing organic inputs, compost, into key phases in the rotation. And I've just given myself a little note there to remind myself that at the Brooms Barn site, because we grow sugar beet in the 7-year rotation, we effectively have a rotational ploughing system, which I know that my other presenters are really interested in as well as a potential solution to reach a compromise with some of the drawbacks, if you like, of reduced tillage. So my last couple of slides before I hand over to Jonah are to introduce some of the results. We've only been going for 7 years, so we're effectively in the transition stage from a ploughed system to a reduced till system, if you like. But the results we're seeing in that transition phase support the literature in that we're seeing a decrease in yields in the reduced till plots, primarily through poorer establishment of the crops. But importantly, that yield penalty of the conservation agriculture or the reduced tillage system is dependent on what's happening in the rest of the system. And this just illustrates that there's an interaction with site and an interaction with preceding crop. I'm not going to talk about the detail, but just want to introduce the principle that if somebody asks, does reduced tillage reduce my yields of wheat? The answer will always be, it depends. It depends on your soil type. It depends on what else you're doing in your system. And the important principle is the real gains and benefits are to be achieved through the layering of these practices and the addition of these regen ag principles. Taking any one in isolation may well not lead to overall benefits. And that's just another example of the kind of results we're getting. These are initial results, so I'd ask you not to share these, please, but the previous slide is published. And the graph on the left shows the impact of compost on spring barley yields, but it also shows the interaction with tillage. So we know that we get reduced barley yield when we reduce the tillage, but that reduction is mitigated by the application of the compost. So we're seeing a greater benefit of the compost to the spring barley when it's in a reduced till system. And these are exactly the kind of benefits and findings that we want to be delivering and making available to the community. And the one on the right there is just an example from biodiversity and natural enemies of pests of oilseed rape. So the little wigwam you see there is collecting parasitoids emerging in a winter wheat following an oilseed rape crop. So those are eggs that have been laid in the previous oilseed rape crop, and those parasitoids are now emerging and will control the pests in neighbouring oilseed rape fields. And we know that in a zero-till system, in a ploughed system, we destroy those eggs and we destroy those pupae, but in a zero-till system, we retain that benefit for the following crop. So again, it's just a flavour of the kind of work that we're doing, the importance of monitoring interactions of practices on multiple outcomes over time. And we're going to use those data to help navigate some of this complexity. And at that point, I'm going to stop and hand over to Jonah. Thanks, Jonah.
Hi, everyone. I'll just get to my screen. So hopefully you can all see the presentation. And I'll lose you while I look at this. So, hi, everyone. I'm continuing on from John and thinking about some of the results from the LSRE as well. I'm going to start off with sort of Regenag and 5 principles. So John touched on it as well, but we've got don't disturb the soil, keep the soil surface covered, keep living roots in the soil, grow a diverse range of crops, and bring grazing animals back to the land. So we can see soil is in 3 out of those explicitly, and then it's in the other 2 sort of implicitly. So it's the real foundation of this regen ag movement, etc. Growing a diverse range of crops then feeds into it through root architecture, rooting depth and things, and bringing grazing animals back to the land. Feels like it's part of enhancing these rotations where we're not growing crops to improve or enhance the inputs that are coming down. And we might also include soil amendments under this sort of umbrella of enhancing these rotations. So taking these then through the lens of the large-scale rotation experiment. So John's given us a really nice overview of that. And I'll also touch on these experiments that John gave us a glimpse of as well. So starting with don't disturb the soil, I've got managing tillage and ploughing responsibly. So like we said, there's some compromise here. The responsibility nature is that sometimes we're going to have to till just as part of rotations, as part of systems, but doing that in a responsible way is what we want to try and find out. And from the LSRE, so I'm just presenting the first 3-year data because I haven't had a chance to look at the most recent sampling because it's fairly recent. But we can see there's an effect of tillage across the treatments on soil organic carbon. So this is change in % carbon. Conventional till has little to no effect, but slight negative. Zero till has positive effect. And when we think about each site, so Broomsbarn, the sandy site, has a slightly bigger decrease in carbon than the sort of zero at Harpenden, but they both have this positive effect with zero till. Then we capture quite a few of them with this next treatment. So this is the amended treatment, the nutrition treatment, where we've got cover crops and compost. Sorry, I meant to have a laser pointer, not a pen. There we go. And this covers sort of keeping the soil surface covered, keeping living roots in the soil, and bringing grazing animals back to land through that circularity nutrient cycling if we extended it to this compost effectively, because we're not always going to have livestock available, etc. But what we can see is that we've got this effect of amended treatment versus the conventional treatment in terms of the carbon, and we can bring in the 4th principle, looking at the combination of the 2. Sorry, I'll just hide that, where we've got this additional effect effectively with the zero till and amendment having a greater increase than conventional and zero till. When we think of when we think of growing a diverse range of crops, that would sort of fall under the umbrella of our rotations, effectively. Like John touched on, we've got our 3-year rotation, 5-year rotation, 7-year rotations. But we don't see a clear pattern here necessarily that we might think, okay, going from 3 to 5 to 7, do we get an overall increase in them? We get a sort of mixed picture. And that's, like John said, there's lots of complexity within this dot effectively. And what we can see from the sites, from the difference just between the sites, the 3-year is being sort of drawn up by the effect of Broom's Barn. Within this data, the 3 years basically had its full cycle. We've had about half of this and just under half of this cycle, so the longer cycle. So there's more to tease out there, and hopefully we'll get some more answers with the next set of results from the 6-year data. So that's where I'm going to finish on the LSRE. I'm just going to bring us to the Woburn and Highfield data. So these are the plots that John showed, where we've got this difference in SOC. The treatments are down here, but largely the black treatments are arable, and these are lay grass rotations. In Highfield, we've got grass, so old grass, reseeded grass, lay, arable, and a bare fallow, which was kept free of vegetation and routinely ploughed to see how quickly carbon could be driven off a grassland effectively. So like John said, we've got sort of sandy soil at Woburn, 13% clay, Highfield, 28% clay, so higher. When we consider their textures, we get a more balanced picture of what's going on with the carbon and the carbon status of the soil, because the Woburn site looks unlikely to ever reach the carbon values of Highfield. You know, it's doing a similar thing to the lays, but it's not reaching the same. But we get quite similar SOC over clay for the arable treatments. Not diagnostic necessarily of an arable treatment, but interesting that they were very similar here. The lays are able to bring it out of this, say, degraded state. And from a regen point of view, this is a grazed lay treatment before this line, where we're seeing sort of this increase almost up to the good line. So that seems quite a positive effect. And then it switches to sort of chemical fertilizers, and you know, it goes down a bit, but not as far as the arable. And here the lays are mitigating sort of that loss going down to the arable system. And we get a better appreciation for this degraded state of the bare fallow, because it's got a similar carbon to Woburn, but it's that much worse effectively because of its clay content. Just to move through, this is my last slide effectively, and this is just a couple images of, or a couple of videos of 2mm aggregates from each of these treatments, so the grass and the bare fallow, and showing sort of the poor spaces and the poor connectivity, how much greater it is in a grassland versus the bare fallow where there's little to no connectivity. So that's me done. Thank you very much. I'll pass over to Jenny.
Right, thank you. I'm just going to share my screen so you can all see my presentation. There, hopefully that's working for you all. So as John said, I'm going to be talking about a little bit about our regenerative agriculture project that we do at the Allerton Project, slightly smaller scale than the many plots of the Rothamsted version, but we farm it in a more farmer-friendly kind of practice rather than a research experiment. So that allows us to look a little bit about the management issues of these kind of systems and the context, as John touched on, and the consequences of what happens when it doesn't quite go to plan. So that's what I'm going to focus on in this talk. But first of all, just who I am and who we are. So the Allerton Project is an education and research trust. Its overall aims is to combine productive farming with wildlife conservation and to research the interactions between farming, wildlife conservation, resource management, so that's where the soil science element comes in, and the environment. And my job on the Allerton Project is to look after that research and help shape its direction. And then we disseminate the results to farmers, policymakers and others. Now, the agricultural sector has a number of challenges at the moment. You see headlines like these about the degradation of our topsoil, and then you have the added impact of the recent extreme weather events, possibly brought on by climate change, with extreme flooding events, extreme drought effects now we're currently going through, which is really adding pressure onto the agricultural sector, and partly why they're looking for not just to improve those degraded soils, but also to look at improving their overall system resilience, their business resilience and their soil resilience for these unexpected events. And to add on to that, climate change is a pressure. Agriculture accounts for 14% of global greenhouse gas emissions and are about 3% of anthropogenic CO2 emissions, but a massive 68% of anthropogenic nitrous oxide emissions. So there's a lot of pressure on farmers to become carbon neutral, particular pressure on farmers because they're in the unique position of being able to grow crops which can actually trap that CO2 from the air, bring it down and place it back in the soil. They're also the custodians of a large swathe, about 2/3 of our, our UK soil habitat. So it's their job to look after it, make sure those soils don't become degraded or become improved. And that's another reason why people are looking to these kind of systems to help that. So this is our farm. Most of these fields have trials on it of some kind, but I'm going to focus on the ones that I've highlighted on this map today. The type of trials we do, we have the Syngenta trial, which is the regenerative agriculture trial I was talking about. It runs across 5 fields, and it's a long-term trial where we take different approaches to farming and just see how it works over many, many years. But we also have shorter-term trials where we look at one specific problem. And it's this combination of these short trials where we look at specific things like adding biochar, adding biostimulants to the soil, and these long-term system approaches means we can understand things in context, pull all these findings together and try and have an understanding of how what we do might impact other areas of our farming. And that's why I find it so interesting to work here on all these different projects. I'm going to start with introducing you to the conservation agriculture project that we have at Allerton. We are the Loddington site, so we're on a heavy clay soil, but there's a sister site over in Lenham, which is on a nice sandy soil. So similar to John's experiment, we realised that the context was very important here, and that looking at these kind of approaches on just a heavy land was not going to be enough, and it needed another site on a nice light land so we could understand the context of the soil type with what we were doing to the farming. And then there's 3 different systems. So there's a conventional, where we use full inversion ploughing. There's a sustainable system where we use non-inversion but semi-deep cultivations, and then there's the direct drill system, which sometimes includes light cultivations depending on the crop and the season, what we're trying to achieve. So we have 3, I think I'd say, approaches rather than regimented systems to farming, and they each run across a field like this. So this is one field split into our 3 treatments, and this is replicated across 5 fields on our farm. And here, I'm not going to go into these results in great detail, but these are the results from our first 5 years of this project. And you can see, as John was saying, context is very important here, because does your yield decrease when you take on these regenerative agricultural principles? Well, on our heavy land, yes, it did, but on the lighter land in Lenham, no, maybe not so much. But overall, our profitability wasn't that badly impacted, because moving machinery through heavy clay soils takes quite a lot of energy, time, fuel. So actually, by reducing those kind of costs, we can actually protect our overall profitability on the farm. So this project didn't just look at the environmental impacts of what we were doing, but we also tried to understand the profitability of running these different systems on different types of land. But what I really want to focus this talk on is what we learned along the way. Because we are a commercial farm and we try and farm using normal equipment in normal ways, we can come up against these unexpected consequences, which I find possibly the most interesting for our research, and certainly for the visitors who come here and want to learn from our trials, possibly the most interesting for them too. I'm going to focus today on the weeds, the weather, and the nitrous oxide, and how those three things impacted our work. So firstly, this is some research actually by someone from the weeds group at Rothamsted, David Comont, who's part of the blackgrass research group. So it was the BGRI network who go around, and they're currently doing it now, I think, mapping fields and looking at blackgrass. And we got him to come and look at our long-term Syngenta trials. And what he could show us was that when you don't plough, you do tend to run into a weed burden, especially if we've had lots of winter crops. We haven't had that period before spring to control it with herbicides. You start building up quite a high weed pressure. And for all that ploughing isn't great for protecting our organic matter and for protecting our soil biodiversity and health and structure, it is really good at controlling weeds. And that's why, one of the many reasons that I've been talking, as Jon Storkey said, about the idea of rotational ploughing, whether ploughing infrequently but to really control the weeds is an option in these regenerative systems, because we do have to consider these problems when we're trying to put in place these manageable systems. And as I said, at the Allerton Project, we have lots of little specific trials looking at one particular thing. And this one is one of our biochar trials, which is in partnership with the University of Nottingham. The whole idea of the trial was to look at adding biochar to soil, seeing what it did to pH, soil bulk density, soil carbon content, those kind of things. But what we found was where we added the biochar in a nice, neat rectangle along our field, we got a lovely, neat rectangle of blackgrass coming up. So again, David Comont was very happy to come along and map that for us, and he could show us that we did seem to have a higher pressure of blackgrass coming up just where we'd added the biochar. Now, this is where it's those building of different management that causes this problem. So because we were a regenerative farm, we top dressed the soil with the biochar, and then we didn't incorporate into the soil. We then sprayed, we drilled straight into it, we sprayed our pre-emergence herbicides while this biochar was still freshly applied and all on the surface. And we think what probably happened was it absorbed a lot of that herbicide and made it a lot less effective at killing and controlling the weeds. And what we got was this unintended consequence, was an enormous patch of blackgrass. So yes, maybe biochar has some benefits, or is possible to be used on fields in certain ways, but maybe not in one of these regenerative systems just on the surface with direct drilling into it if you're trying to control your weed pressure. So another unintended consequence there. And then we move into the weather. So the results we showed you with the profitability of our regen ag system was shown after the first 5 years of the project. There was some bad weather during that, but it really started after those first 5 years in about 2023, when we had Storm Babette kicking off the season, just when we might have been wanting to drill our winter crops. And then the weather continued all through the autumn, all through the winter, all through early spring, and we couldn't get on any of our fields. They weren't travelable because we're a heavy clay site. Infiltration is about, maybe if you're lucky, 4 millimeters an hour, and when you get 63 millimeters of rain in a day, it's going to be a long time before that field percolates all that water away and you're able to travel again on that field. And when you're getting more and more rain on top of already soaked soil, you're not going to be able to get on. We found that when the weather did this, there was actually a benefit to the fields, the plots that had been ploughed, because we'd aerated them, we'd lifted them, we'd increased the infiltration rate. the little graph you can see there on the right is the compaction measured in those different plots. And you can see that there is, particularly in the first 20 centimetres, less compaction in that ploughed soil. And that meant the water could infiltrate just that little bit faster through those ploughed plots. And what we got was much better establishment. So when farmers are thinking about what they can do to make their soil and make their business more resilient, make sure they get crops in when we're getting weather like this. Some, certainly on heavy lands, are starting to think, maybe it's ploughing, maybe I need to go back to this in order to make sure I can get a crop in. However, if you look at that line running through the middle of that field, you can see that it's not without its risks. So where we had the direct drilled soil on either side, there isn't such a deep channel running through it, but where we had that fluffy soil that had been worked, was broken up into smaller aggregates, was more mobile, it's really dug a deep channel of soil erosion. So yes, maybe it can infiltrate the water more, but in turn it is more prone to erosion risks. So trade-offs there as well. And lastly, when we get events like we had in 2023, 2024, and to a certain extent the beginning of last season as well, you get another unintended consequence, particularly on heavy land like ours, which is nitrous oxide. Nitrous oxide is preferentially produced in anoxic environments. So when you have a direct drilled field and there's very little pore space, it fills up quickly with water, and that water then becomes anoxic very quickly, and suddenly all the nitrogen in that field is going to be released as nitrous oxide. This picture is of one of my long-term experiments where we're just comparing ploughing every year with direct drill. We are very, so this isn't farmed as a farmer would, as a systems approach, but more as just a comparison between what happens if you plough, what happens if you do not move the soil at all. And what we found is after those seasons of bad weather, we ended up with no crop establishment because all the seed was sitting in water for far too long. And we couldn't get in to establish a crop for many, many months. So that whole season, it was just being battered by the rain, and nothing was growing in it in those direct drill plots. And we'd been watching slowly as the soil structure improved over the last 13 years that this experiment's been going on, and suddenly the soil structure started plummeting. And when I went to take some measurements, I found that there was a real increase in nitrous oxide. So the nitrous oxide you can see here in the direct drill plots was enormous compared to when we had the plough plots, or even when we alleviated some of that compaction with subsoiling. Now, we had already previously looked at this on a specific compaction experiment where we'd driven up and down many times with a tractor. I think it was about 8 hours the farm manager spent in a tractor trying really hard to compact the soil for me, and he did a great job. And we could see that there was this slight increase in nitrous oxide when we looked in the winter, when the soils were saturated and wet, compared to when we looked in the summer. But generally, CO2 was still the biggest impact on the emissions. But when I saw this result, which happened without any artificial compaction, it was purely the result of direct drill plots that hadn't managed to establish a crop in a really bad year of weather on a heavy soil. It became very real for me that this is a genuine problem that farmers might find in the regen ag system when farming these kinds of heavy soils. So again, unintended consequences and trade-offs we have to think about. And another reason why occasionally cultivating in a rotation might be considered the best option in terms of soil health, crop establishment, and even climate change. So in summary, regenerative agriculture has many trade-offs, and as Jon has said, it's very important to consider the context when making decisions. So thinking very hard about what kind of soil you have, what you're trying to achieve, what kind of crop you're growing, and even what the weather might do. I think we have a role in research to support the education and help with the decision making that these farmers have to make. And there is movements now in agri-tech to also help support these decisions, but in the end, the decision still has to come down to the farmer. So there is need for support from the industry, the government and consumers to help farmers make the right choices on this journey towards improving our overall soil health. Thank you very much.
That's great. Thank you all. Thank you all for your presentations. That was really interesting. So we've now come to the Q&A section of the webinar. So I'm going to invite all our speakers to join me back on camera. Just a reminder to our audience that if you have any questions, please submit them via the questions box, and we'll try and get through as many as possible. So just to kick off, I'm going to take Chairman's prerogative and ask a few of my own. Jon, thank you for the outline of the Broomsbarn Harpenden rotation experiment. I can see that that's going to be a really valuable resource for us to understand the impacts of these sort of farming systems. So I think you said you started in 2017, 2018. So I was just wondering, well, firstly, for you to date, what are your headline messages or the key things that you found from that work?
Okay, so the first one is that some of the unintended consequences that Jen has referred to can be mitigated by other practices. So, for example, the poorer establishment on the no-till plots can be mitigated with increasing the soil organic carbon, or increases the infiltration. So it's layering practices. And then the second thing is, you're right, that this element of time. So what I've effectively presented is the transition phase, but we are seeing the regenerative plots get gradually better. So the difference between the zero till and the ploughed plots in terms of yield is decreasing with time. So we need a long-term monitoring approach to this. So I think the temptation is to try something for a couple of years, see it doesn't work. For example, the slugs might go crazy, or you might get poor establishment, or you might get weeds, and then give up. But I think the value of this experiment is to provide the evidence base to give people the confidence to stay the course, to say, well, if you adapt your system in this way, you can still retain those benefits and make it work.
Yeah, I think it really highlights the benefits of those long-term trials. Do you know how long? I mean, obviously your ambition probably will be to keep it going for very many years in the future, but do you know how long you've got secured for that piece of work at the moment?
So we have the advantage, as Georgia referred to at Rothamsted, of being supported by something called the Laws Agricultural Trust, which is a charity that also maintains the long-term trials like Broadwalk. And our ambition is to integrate this new trial within that set of experiments such that it has that long-term security. But you're right, it is really important. Our statistician who designed the experiment has worked out that I think all the rotations will go around and all the wheels and cogs will turn, and it will get back to its starting point in 120 years. So that's our
Fantastic.
That's our ballpark.
I like that answer. 120 years, brilliant. Also, to pick it up, I liked what Jenny, your comment on the unexpected consequences. It's always interesting. We have sometimes these assumptions of what we might find. So Jon, if I may come back to you again, was there anything in particular that you found with that work that you really weren't expecting, that you hadn't anticipated?
It's not necessarily an unintended negative consequence, but looking at the data recently, the plots that we apply compost to in the zero-till system, they have less trash in the spring. So that by the trash, the stubbles and the crop residues. So in the zero-till systems, they're left on the surface. They can sometimes be a problem in the following spring. Well, they can support slugs and diseases, and they can also be a problem in drilling the following spring. The compost, where we had the compost, the amount of trash is decreasing. So that was an unexpected benefit of the compost, that we're effectively feeding the microbiology and the soil biology, which is decomposing and removing those crop residues at a faster rate.
Yeah, okay, fascinating, thank you. Jonah, I really like the image that you put up showing the aggregates from the fallow and the grassland. I thought they were fascinating. What I was wondering was, does that, with that technology, do you just get that image? I mean, the image is great. I was wondering whether you were able to extract from that any sort of numerical value of the pore size.
Yes. So it's not my image. I have taken that from sort of Andy Neal and others' work, but yeah, they definitely have that data and can look at then building models on the porosity of the soil and things like that, and looking at it in terms of organic matter, moisture content, things like that, to draw out these relationships and get better understanding of what's going on in those systems.
So are you managing to use that technology on sort of the experiment that Jon was talking about?
I think Jon is possibly nodding. Yes, as in yeah. So Jon will have more information of whether it's been used yet, but I think it would be great to look at different rotations in the LSRE and think about differences in terms of length of time since ploughing in sort of the rotational plough, or the difference between no-till and tillage, different cropping sequences as well there in terms of the roots, whether we've added amendments or not. So to sort of dig into some of those unintended consequences and see whether we can link any of that together, basically, through the soil structure in that way.
Yeah, and that's the plan we have. So the opportunity we have on the Brooms Barn site is to answer the question: if you plough infrequently, how much of the good in terms of that improved soil structure and soil aggregate stability and porosity is undone by a single ploughing event?
Yeah.
And I think that's a really important question.
I think it's interesting there as well, because it's the sandier soil. So, you might disrupt the structure more easily. And yeah, depending on, there might be differences there in the recovery, but also then on the clay soil, you have different structural properties and compaction issues as well. So yeah, it would be interesting.
And do you see a correlation? What sort of correlation do you see between those sort of measures of pore space and other indicators of soil quality?
That I need to look at the results again a bit more, but I'm sure that there is a positive relationship with organic matter. So, I mean, just from that contrast of the bare fallow to the grassland, we definitely see that contrast reflected there. And I think also then moisture content and infiltration rate will follow soil porosity in terms of the air content of the soil as well.
Okay, that's great.
To add on that, we did some research back when I worked with the British Beet Research Organisation, and we were looking similar at those pore spaces and the connected pore space, and what we found was really good links with seed establishment, because it's that water and air movement through the soil, particularly in the topsoil, which was crucial, and which I clearly wasn't getting in one of my very compacted experiments. So it's again that soil structure that's optimal for allowing seed establishment, which leads on to all kinds of things for good soil health. If you can get something growing in there to start with, it's a very good start.
Yeah, and Jenny, I like, well, the photo you put up of the soil, the sort of the water running off the fields in that sort of heavy rainfall. It is pictures that really make us sit up. When you see soil running off the field, that is the last thing any of us want to see. Looking from the results that you presented, it kind of seemed that there were some potential benefits of ploughing in terms of infiltration and sort of reduced nitrous oxide, which is really, when we talk to farmers about what to do for soil health, we routinely all say minimise soil disturbance. But I was wondering whether compaction was an issue for you on those other plots.
Yeah, I think because we do lots of different studies, on that study it was plough versus direct drill, and on that year ploughing gave us that chance to get a seed bed established, whereas it didn't because But as Jon says, if we had got to the point where our organic matter had built up more, maybe there would have been a different story for our directorial fields. But in those 5 years, it hadn't reached that point and it was a problem. But on another site, we had compared subsoiling with ploughing and with direct drilling, and in that instance subsoiling came out as that happy medium which lifted that compaction but didn't do full inversion tillage, so we didn't destroy those habitats for the mycorrhizae, you know, we didn't completely we didn't reduce the carbon storage that we've been building up there. So I think there are always other options to consider but the overall story is compaction needs to be dealt with. We can't just ignore it and hope that we can grow our way out of it. We need to deal with that before we create more soil degradation. So.
Yeah, I think that's a really good point, that potentially if you've got compaction in your field already, that you might need to address that, and tillage might be the best way of doing that. It is quite a complicated message. I mean, if you were talking to a farmer on similar heavy land to you've got, what would you be talking to them about in terms of whether they should till or not to till?
I think my message is always to take your spade out and look and think about what it is you're aiming to do there, and think about the least intrusive thing you can do to get your soil to the next step, and build that slowly along your journey. Just throwing away your plough and saying I'm never going to do it again from this point clearly wouldn't work. We need to slowly move our soil to a better state where it can be of higher organic matter and it can be slightly more resilient when this rainfall comes. But to do that, you might need a slower journey than we hoped, unfortunately.
Yeah, thank you. We do have one question in the chat that's slightly specific about PhD funding, so I think we'll pick that up offline and we'll come back to that person after the webinar. Just to kind of round us off, that we've all seen that massive increase in interest on regenerative agriculture and regenerative farming from farmers, and quite a few of the farmers definitely that I know are sort of experimenting on their farms. The experiments that you've described, you've described in your presentations today, are obviously not the sort of experiments that you could possibly do on a farm, those hugely in-depth experiments. But if you were talking to a farmer who was sort of embarking on that journey and keen to experiment on their farm, I just wondered whether you had any pointers or advice to that person. Jon, I might come to you first, but then Jenny and Jonah, if you want to chip in as well.
It's a really good question, because I've just been away at a meeting where I've been thinking a lot about how we bridge this gap between the evidence that the likes of Jenny and Jonah and myself are collecting and making that accessible and relevant to individual farmers' decisions. So it's a really good question, but it's one I find difficult to answer, because you need there's some kind of intermediary steps that you need. I think Jenny at GWCT are much better at this than we are, in terms of making all that evidence accessible and in a form that they can say, okay, given what they found at Loddington, given what they found at Rothamsted, if I do this, what can I expect to see based on my soil texture and what I'm doing? So it's not an answer. I think it just highlights the challenge that we've got, and places like Groundswell are really good arenas for bridging that gap. So I think Jenny, you'll be there, and we'll certainly be there as Rothamsted. So it's seeking out people at events like Groundswell, I think, would be my answer.
Okay, good one. Jenny or Jonah, did you want to add anything to that?
I can say that the Allerton Project will be at Groundswell, and we're always happy to talk to people wanting to do trials on their farm. And yeah, there are various resources and people you can talk to for some support for that kind of thing.
Yeah. Okay. No, thank you. And I think this webinar has been well timed just before Groundswell So yourself, you're there, Jenny. I think Rothamsted are there. The British Society of Soil Science are there as well. So for those listening, please do come and find us all. But I think we'll draw that to a close now. So I'd like to thank you all. So yeah, on behalf of the British Society of Soil Science, I'd like to express our thanks to all of you today, to John, to Jenny for coming along to present, and also Jonah and Georgia from Rothamsted. Thank you all, everyone, for attending. You will find a quick feedback survey when you leave the webinar, which would be really grateful if you could take the time to complete. The recording of the webinar will also be available after the event on our YouTube channel. And our next webinar is scheduled to take place on the 23rd of July and to be led by Arcadis. So keep an eye out on our website and follow up the email for more details. So hope to see you at future events and at Groundswell if you're there. And in the meantime, thank you again and goodbye.